If you are watching this right now, and you know your blood type is O positive, what you are about to hear may change the way you think about your own body for the rest of your life. O positive is not just a label on a hospital wristband or a line on a donor card. It is part of a genetic story that has survived for tens of thousands of years. It has been passed silently from body to body and from generation to generation, long before anyone had names for blood types, and long before anyone understood that blood carried hidden biological signatures at all. In recent years, something remarkable has happened. Scientists working in population genetics, ancient DNA research, and molecular biology have begun piecing together a story that had been sitting quietly inside human chromosomes the entire time, waiting to be read. This is the story of where O-positive blood may come from, why it became one of the most common blood types on Earth, and why the answer reaches back into a part of human history that most people were never taught in school. Before we go further, if hidden layers of human ancestry and the science of survival fascinate you the way they fascinate us, take a moment to subscribe. This is the kind of careful research that often gets buried under clickbait, and we are here to bring it to the surface properly. Now, let's begin. To understand where O-positive blood comes from, we have to go back to the year 1901. That was when an Austrian physician named Karl Landsteiner sat in his laboratory, mixed blood samples together, and noticed something strange. Some samples clumped when combined, others stayed smooth. That single observation opened the door to one of the most important discoveries in the history of medicine: the blood group system. Landsteiner identified 4 groups: A, B, AB, and O. For most of the 20th century, many scientists assumed that type A was the original ancestral form, and that the other blood types branched off from it. At the time, that seemed reasonable. Type A appeared widely across Europe, and early blood research was concentrated in Western laboratories, studying mostly European populations. But that assumption was based on limited data. Once genetic sequencing technology improved over the last 2 decades, researchers returned to the actual gene involved, and the older theory no longer held up. That gene sits on chromosome 9. It produces an enzyme that determines whether certain sugar molecules, called antigens, are placed on the surface of red blood cells. If the enzyme adds one kind of sugar, a person has type A blood. A different sugar produces type B. If both are present, the result is type AB. But type O is different. Type O exists because of a mutation that disables that enzyme almost completely. With no functional enzyme, no A or B antigen is added to the surface of the red blood cell. When researchers compared indigenous groups, ancient remains, and modern populations side by side, the global pattern of type O began pointing again and again toward Asia. This is where the story becomes more specific. And this is not just speculation. It is grounded in population genetics research that has been building for decades and has become more refined in recent years as sequencing tools improved. Before European contact reshaped the genetic landscape of the Americas, many indigenous populations showed extremely high frequencies of type O blood. In some communities, researchers found levels approaching near total saturation, meaning almost every person tested carried the O allele. That does not usually happen by chance. In population genetics, when a trait becomes nearly universal in an isolated group, it often points to what is called a founder effect. That means a small ancestral population carried the trait, and because their descendants remained relatively isolated for thousands of years, the trait spread through the lineage with very little competition from other variants. When scientists trace Native American ancestry backward using mitochondrial DNA Y chromosome haplogroups, and whole genome sequencing. The trail consistently leads into Siberia, into ancient Northeast Asia, and into a population bottleneck known as the Beringian standstill. This is not a fringe theory. It is one of the best supported models in modern anthropological genetics. It describes a period during the last ice age when a relatively small human population became isolated in Beringia, the land bridge that once connected Siberia and Alaska, for possibly thousands of years before their descendants moved south into the Americas. During that long period of isolation, the genetic traits of that founding population, including their blood type frequencies, became established and were then carried forward into the populations that followed. Here is the part many people never hear. Type O was not only historically dominant in the Americas because of that founder migration, it also remains one of the most common blood types across large parts of Asia today, even though the exact percentages vary by region. In parts of Southeast Asia, including places such as the Philippines and Vietnam, O frequencies remain very high, often reaching or exceeding around 40% of the population. Across East Asia more broadly, type O is often among the leading blood types, alongside type B, with the balance shifting from country to country. The key point researchers keep emphasizing is this: Asia is not just one region among many when it comes to type O. It appears to be one of the largest genetic reservoirs of the O allele on the planet. Multiple migration events out of that reservoir, spread over tens of thousands of years, helped carry high O frequencies into populations as far apart as the Arctic and the southern tip of South America. What makes this even more interesting is that scientists do not find just one identical O mutation everywhere. They find several different ways the ABO gene can be altered so that the enzyme loses its function. In other words, there are multiple molecular paths that can lead to type O blood. Even more importantly, the O variants that are common in East Asian populations often carry distinctive nearby genetic markers. These markers act almost like signatures in the DNA, allowing researchers to trace specific versions of the O allele backward, through time, and across populations with a level of precision that was not possible even 15 years ago. And when they follow those signals, they often lead deep into the genetic history of ancient Asia, tens of thousands of years before recorded history began. But the story does not stop with migration. It also touches ancient interbreeding, disease survival, and one of the strangest blood conditions ever discovered. Long before modern humans spread across Asia, the continent was already home to other human groups that had lived there for hundreds of thousands of years. When modern humans moved into these regions over the last 50,000 years or so, they did not simply enter empty land. In some places, they encountered earlier human populations, and ancient DNA research has confirmed that interbreeding did occur. Researchers studying blood type genetics believe those ancient encounters may have contributed to some of the blood-related genetic patterns seen in Asia today. This remains an active area of research, and scientists are careful not to overstate the case. But the timing is close enough that it continues to attract serious attention. Once type O is combined with the Rh factor, the marker that makes blood positive or negative, we arrive at O positive. And O positive turns out to be statistically dominant across large parts of Asia and across many populations connected to Asian ancestry. That raises the next question. Why did this blood type spread so successfully across so many generations, environments, and migrations? One of the strongest explanations in the scientific literature involves disease. For most of human history, the greatest threat to survival was not war or predators. It was infection. Parasites, bacteria, and epidemics killed more people across history than almost anything else. And blood type appears to influence how the immune system interacts with certain pathogens. Multiple studies have found that people with type O blood can show different risk patterns for certain forms of malaria, some bacterial infections, and even some viral illnesses. That does not mean type O provides a universal shield. It does not. But over many thousands of years, even a small survival advantage can change a population dramatically. If one blood type gave ancient people even a slightly better chance of surviving childhood during a major outbreak, that advantage could spread through the gene pool over many generations until it became very common. Many researchers consider this one of the leading reasons why type O reached such high frequencies in parts of ancient Asia, and why those frequencies were later carried into populations across the Americas and the Pacific. But genetics almost always involves trade-offs. Type O blood has also been linked in multiple studies to a higher lifetime risk of certain conditions, especially stomach ulcers. Research has repeatedly found connections between type O and greater susceptibility to Helicobacter pylori, a bacterium that can thrive more easily in the stomach lining of people with this blood type. That creates an interesting question. Why would the same blood type that may have helped ancient populations survive some infectious threats end up increasing vulnerability to other conditions thousands of years later? There is no simple answer yet. Genetics is full of trade-offs. A trait that helps in one environment can create risks in another. Then there is one of the strangest discoveries in blood science, the Bombay phenotype. It was first identified in 1952 in the city then known as Bombay, now Mumbai, and it remains one of the rarest and most medically important blood conditions ever documented. People with the Bombay phenotype may appear to be type O on routine testing, but at a deeper genetic level, they are different. They lack a foundational molecule called the H antigen. That molecule normally sits beneath the A and B antigens and acts as a base for the entire system. Without the H antigen, a person cannot produce A or B antigens properly, even if other parts of their genetic makeup would otherwise allow it. Their blood looks like O on the surface, but it is structurally different. Most importantly, people with the Bombay phenotype cannot safely receive ordinary type O blood. They usually need blood from another donor with the same rare condition. This makes it one of the rarest and most medically significant blood profiles on Earth. The condition appears most often on the Indian subcontinent, at roughly 1 in 10,000 people, compared with around 1 in a million in many other regions. It is a reminder that even within what seems like a simple blood type, there are hidden layers of genetic complexity that standard hospital typing does not always reveal. And once again, many of those deeper layers point back to South Asia. When we step back and look at the bigger picture, the evidence from migration, disease, trade-offs, and rare blood conditions suggests something important. O-positive is not just a medical label. It is a historical record written in biology. It was shaped by survival, disease, movement, and inheritance over immense stretches of time. For most of the 20th century, scientists had to infer ancient blood type patterns indirectly by studying living populations and cautiously projecting backward. That changed over the last 15 years as ancient DNA technology advanced. Today, researchers can recover readable genetic material from human remains that are 5,000, 10,000, and in some remarkable cases more than 30,000 years old. They have sequenced the genomes of people who lived during the last ice age, people who saw the earliest shifts toward agriculture, and people who lived near the beginnings of the first civilizations. And every time blood type markers are examined in these ancient genomes, they add another piece to the same, larger story. Ancient DNA studies suggest that type O was already widespread in East Asia at least 30,000 years ago, long before agriculture, long before writing, and long before the modern nations we know today existed. Researchers studying ancient remains linked to the ancestors of Native American populations have found O frequencies that were, in some cases, even higher than those seen in many present-day descendants. That fits closely with the founder effect model we discussed earlier. In early farming communities in China, scientists have also identified O-related variants preserved in skeletal remains, reinforcing the idea that these roots extend deep into Asian prehistory. These roots are no longer just speculative. They are increasingly documented, sequenced, and traced with growing precision. One especially interesting area of research involves ancient trade route populations. Scientists have examined remains connected to some of the earliest travelers and traders who moved through corridors that would later become part of the Silk Road. In some of the oldest layers from those regions, type O appears to have been much more common than it is in many of the same areas today. That suggests the earliest travelers through those routes, before later waves of conquest and migration changed the gene pool, may have carried type O at especially high rates. That means a person alive today with O-positive blood and with East Asian, Central Asian, or South Asian ancestry, somewhere in their family line, may be carrying a biological signature that reaches back to populations whose names are lost to history, even if their genetic legacy is not. There is another migration story that deserves equal attention. Roughly 5,000 years ago, seafaring populations from the region of what is now Taiwan began an extraordinary expansion across the Pacific. Navigating by stars, currents, and inherited knowledge, they moved from island to island across immense distances of open ocean. Over time, this movement helped give rise to the ancestors of many modern Polynesian populations. When researchers examine blood type frequencies in Polynesian groups today, they often find very high concentrations of type O. In fact, Polynesian populations and indigenous populations of the Americas are often among the most O-dominant groups documented anywhere in the world. What makes that striking is that these two population groups are separated by the largest ocean on Earth. They arrived through different migration histories, at different times, by different routes, and yet both preserve the signature of that same ancient Asian reservoir of type O. In a sense, the ocean did not erase that inheritance, it helped preserve it. There are still important questions that researchers have not fully answered. Why does type B reach such high frequencies in northern India, higher than in almost any other studied region? Some scientists think this reflects migration from the Central Asian steppe, where type B may have risen earlier before moving southward. Others think it may reflect local origins or selective pressures unique to that region. There is also the question of why type AB, the rarest of the four major blood groups globally, appears slightly more often in East Asian populations than in many European ones. The leading explanation is simple probability. If type B is already more common in a population, the chance of A-B combinations increases when A and B lineages mix. Researchers also continue to study isolated communities in places such as Taiwan and the Philippines, where blood type frequencies still resemble older Asian genetic patterns more closely than those of many mainland populations. The most common explanation is long-term relative isolation. These communities may have avoided some of the major waves of migration and mixing that reshaped other regions, preserving something closer to an older genetic snapshot. When scientists follow all of these threads far enough, they tend to converge on the same broad conclusion. The story of O-positive blood is not random. It is deeply connected to ancient population history, disease pressures, migration, isolation, and survival. And many of those lines of evidence lead back to Asia as a major center of that story. There is one more layer that ties much of this together. It comes from the parts of DNA that pass down along largely unbroken lines. The Y chromosome passed from father to son, and mitochondrial DNA passed through the maternal line. These genetic systems preserve deep ancestral patterns. In East Asian populations, certain Y chromosome and mitochondrial lineages trace back to ancient ancestors who lived across Asia between roughly 30,000 and 50,000 years ago. When researchers compare those ancient lineages with blood type frequencies, a striking overlap appears. Populations carrying some of the oldest, long-standing Asian lineages also tend to show stable and enduring high frequencies of type O. The paternal line, the maternal line, and the blood type patterns often point in the same general direction. On the medical side, modern research continues to find that people with O-positive blood can show slightly different patterns in cardiovascular biology, including differences in clotting behavior. Researchers have also examined possible links between blood type and mosquito attraction, which may matter in regions where mosquito-borne illnesses remain a serious health threat. None of this makes O-positive better or worse than any other blood type. It simply shows that blood type is woven more deeply into immunity, biology, and history than most people realize. So what does it mean if you look down at your chart and see the words O-positive? It means your blood carries a story older than any nation, any border, or any flag. It means that somewhere in your ancestral line, real human beings survived disease, crossed frozen land bridges, sailed open oceans, and passed that inheritance forward until, eventually, it reached you. It does not define who you are, but it does connect you to one of the longest survival stories our species has ever lived.